It's lovely to be here with you this afternoon. So my topic's a little bit different from what you've been hearing earlier on today. I'm going to be taking more of a winery perspective. So my topic is the BRI Winery and the impact that it has on your vineyard. Some of you have probably been to the BRI Winery, but many of you probably haven't. So I wanted to give you a bit of an overview about who we are and what we do. So the winery itself is based on the NMIT campus, along with plant and food and a number of other viticultural and winemaking businesses. It was opened in 2020, and it was opened to become sort of a hub for scale-up sized research, both BRI-led and also commercial research. You might be thinking, well, what interest is that to me? It's a winemaking facility, right? Well, actually, 70% of our trials are actually related to vineyard changes, new sprays, a new way of managing your canopy. I thought I'd do a quick overview video of some of the interior of the winery and some of the things we do in order to run trials at the BRI. So, like I said, we're opened in 2020. We're based at the back of NMIT, and we're a Green Star 5 rated building. So that means that we run on very sustainable practices, such as we harvest our own rainwater, we have solar panels. We can customise trials for wineries, for agrichemical companies, viticultural consultants, and we're an end-to-end winery, which means we complete everything from receival right through to bottling. So I work with customers to design a trial for them based on what they're wanting to get out, what results they're looking for, how we can help them maximise their time with us at the BRI. We have 2 different size tanks. We have very large, 200 litre ferments, large for us, and so that's kind of more scale up again. Most people choose to ferment in our 17 litre ferment tanks. Every single tank is temperature controlled. Every single tank has exactly the same conditions, so that we're making sure that the results are even and predictable, and therefore we can compare results from one tank to another. We have our own little bottling line, and we can run trials for wineries and agrichemical companies of any size. We've got a lot of our own on-site analytical equipment, and we use partners like Pacific Rim, Plant and Food, Hills, for any more technical analysis that we may need. I know I'm the last speaker of the day, but if anyone wants to approach me after this, feel free. I'm happy to talk to you about what we do and give you some more information. So over the last 5 vintages, we've run a lot of trials that have impact on vineyard, both commercial and of our own research. Some of the things we've looked at, as Yuichi has spoken to you about earlier, are things like new canopy management techniques, and if you're going to change those, are they going to impact on your final wine quality? We're running currently a project in collaboration with EIT up in Hawke's Bay, looking at varying shading canopy on Albarino and how that impacts on volatile aromas in the finished wine. We've also dealt with vine nutrition regimes, people trialling different fertilisation techniques, trialling perhaps a new biological spray, and how that might impact on final quality and the yan that comes through into your wine. I'm not going to touch on those today. My presentation today is about how we can help new products be included into the SWNZ spray schedule. Now, I'm probably preaching to the choir here, preaching to the converted. Most of you in the audience are a viticulturist, a grower, a vineyard worker, and you will all know that one of the most important documents that you'll use throughout your growing season is the spray schedule. It's really important to adhere to that spray schedule. It's part of being a certified vineyard. And being certified means that you can actually get your product to market and be able to export your wine. Products are quite regularly added, removed and updated in the spray schedule, and that's to make sure that we adhere to international regulations and limitations as these change from time to time, particularly if you're looking to export into the European market. That's one you've got to keep an eye on quite frequently. So how do products actually get into the spray schedule? As you'd probably expect, it's a very thorough process, and it's aimed at ensuring the efficacy of any spray, whether that being a new chemistry or a new biological product that's used in any SWNZ accredited vineyard. So there's 5 steps to getting a new product included. The first 2 are outside of the BRI kind of realm. The first one is actually getting EPA approval. So that's the Environmental Protection Authority approval to actually bring the new chemistry into the country. The second step is gaining MPI ACVM registration for that spray with a label claim. And that's the label claims that Vaughan and Andrew were just speaking about before when they were talking about things like your spirotetramat. The third step is where BRI can come in and help with providing data to SWNZ to get that product into the schedule. So an agrichemical company must put together a large amount of data to show that the product is safe, to show that it does what it says on the label, to show that it's not going to be producing off aromas and things like that in the finished wine. That data pack gets submitted to SWNZ, they assess it, and they go back to the client and say, yep, your product meets the standards that we require, it's now going to be included into the schedule, or else they say, sorry, it doesn't. Please go back, run more trials, run more experiments, get us more data and submit again. So there's actually 6 ways that the BRI can help providing data for that pack. The first one on the list there is commercial field trials. That's with Chris Ireland, who I'm sure many of you know. So Chris runs the actual applying of the new chemistry in the vineyard and does all of the monitoring and data collection in the vineyard. So if anyone wants to talk to Chris later, hit him up. I'm sure he'll be quite happy to talk to you. Once the grapes come into the winery, that's where we come in. So there's 5 areas there that we look at. 1, we send wine off for spray residue analysis. We look at juice composition when the fruit comes in. We look at the rate of fermentation kinetics, the drop in sugar levels or bricks. We look at the finished wine composition, and then finally we gather a team together to run sensory analysis. Why is residue testing important? SWNZ need to see that at the rate that is going to be stated on the label, and also at 2 times that rate, that there is nil residue left in the wine. So an agrochemical company has to supply data and show that this residue is not there in the finished wine. In the vineyard, the spray is applied at 3 different rates on a commercial white and red variety. In general, we're looking at Sauvignon Blanc for white, Pinot Noir for red, but there are a couple of other varieties people can choose. It has to be sprayed at the commercial rate, 2 times rate, and then at either 5 to 10 times rate that is much higher than what you'd ever see being used when it gets into the spray schedule. Why do they need to have that very high rate, the 5 to 10 rate? It's so when it gets to the accredited lab, we will actually have some residue in that very high rate so that the lab can, and the chemical company can pinpoint at which point there would be nil residue. So the grapes come into the winery. We make the wine according to the best practice commercial winemaking for that particular grape variety. We treat the wine just as it would in a real situation. So the wines are fined like they would be in a winery, filtered and bottled using normal commercial practice. We then send the wine off to an accredited lab who'll run testing, usually with either HPLC, GCMS, to run it against a known standard. Key point is to ensure that whatever this new spray is, it has no impact or very little impact on the ripening of your fruit. For the next few slides, I've pulled up some data from agrichemical trials that we've run either last year or we've just finished running this year. For all of them, I've removed the name of the company involved for confidentiality, but all of this data is data from trials. So you can see here, we're comparing Brix, pH, TA, malic acid, and YAN. They're pretty standard maturity parameters. So some of the things that we're looking for is to make sure that the sprayed fruit is isn't showing lower ripening, isn't showing hugely way out of spec acids, and we're not seeing a reduction in YAN. You can see for this particular trial that actually the sprayed rates, the Brix level actually went up slightly, but really there's no large difference as you can see there. And I have to say, over the last 2 vintages, and pretty much all of the agrochemical trials that we've run, we're not seeing a very big difference between the control and the spray. Fermentation kinetics is very important. We want to ensure that anything that has been put onto the grapes is not going to impact the yeast viability once it's actually in the winery. So how we do this is we obviously we add the same yeast for every single treatment at the same rate, and with our temperature-controlled tanks, we will obviously ferment at exactly the same temperature. We monitor the Brix level daily. We monitor the temperature, we monitor the Brix, we smell the wine to check that we've got no off aromas developing during the ferment. And at the end of the fermentation, we turn this into a graph. It's easier to see it visually. And you can see here that really between the control and each of the rates, there is no difference in that fermentation kinetic. And that's the kind of result that a chemical company is going to want to see. There is no impact of that spray whatsoever on the yeast health. Just like we tested the grape juice, we obviously want to check that the finished wine, as far as chemistry, wine composition goes, is not any different from the control. And you can see here, there's some variability, but if you look across all of that data, it's roughly the same, and that's the kind of result that a chemical company would like to see. We're not seeing huge differences in pH and TA, malic. Glycerol is what adds to kind of mouthfeel and roundness to wine. And you can see with this particular trial, really there's no large differences there that stand out. For red wines, SWNZ also calls for colour and phenolics, or tannins, to be analysed. Very important. As a consumer, the first thing people do when they pour out a red wine is look at the colour. Both at the kind of the density of it and also at the hue. So we analyse, we use a piece of equipment called the Cloud Spec. It has a number of different analyses that it can complete. And then we can compare them against each other. So some of the things we're looking at, the hue. The hue gives us an indication of whether the wine is quite youthful or whether it's aging. If the control wines were more towards a higher number in the hue, it might give us an indication that something's gone a little funny there with the spray. With this particular trial, you can probably see that up here in intensity, they've got a bit funny on the side there, the yellow is rate times 5. And this is really the only one that stands out for this particular trial. And you can actually see there that the intensity of the rate times 5 is quite a little bit higher than the others. This may not actually be a bad thing, it just means actually the wine is actually more deeply coloured, more intensely coloured. So that SWNZ may look at that and actually not see that as a negative, but it is something they would look at. As you can imagine, sensory analysis is a huge part of the data pack that we put together for SWNZ. The last thing you want is your chemical that you're trying to introduce into the spray schedule leaving an aroma or a taint or a fault in your finished wine. We get together a team of winemakers, usually 12 to 15 winemakers gather at the BRI. They've all been trained in sensory assessment. They taste the wines blind and in a randomised order to ensure that there is no effect of placement. It's a randomised test. They analyse the wines for many different attributes, things like acid, different aroma compounds, things like tropical fruit or stone fruit. They look at palate balance, length, complexity. They also make note of any faults or any off aromas they may notice with the wine. We then present this data in a spider plot. And we're hoping, what you're wanting to see is that the control and all of the sprayed samples show no difference or show an improvement. This one actually is a really, really interesting assessment. This was a biological, so we've actually only got control and then 2 rates. They didn't actually need to have the residue. But what we can see here is that at both the commercial rate and the 2 times rate, positive attributes were all greater than the control. So the company with that product, I think we're very happy with that result. Hopefully we'll see that one coming up on the spray schedule sometime soon. Finally, every single step we've taken in making that wine from receival to bottling to sensory is collated into a report, handed over to the client who includes it in that data pack required for the SWNZ assessment. I'd like to take the time to acknowledge Jasmine Howie from SWNZ. She's helped me quite a lot with putting together this presentation. And also Chris Ireland. We work very closely together and he's helped me a lot put together this presentation as well. Thank you very much.